Hello warriors, welcome back to our channel of Curious Minds, and today I am going to discuss with you a very interesting concept on blood groups. Let's understand how does various blood group arise. So here is in front of you. So to answer this question, we are going to take you back that how the different blood groups are introduced. So as you know that in the blood there are blood cells and plasma. In blood cells we got RBCs, we got WBCs, we got platelets. But for blood groups we are going to focus on RBCs. As RBC bears certain antigen, so let me put an RBC in front of you. This RBC shows various antigens on its surface. And depending on these various antigens, we got different types of blood groups like ABO, RH, we got a Duffy antigen, we got KID antigen, we got Lewis antigen, and we got MNS antigen. Now these are various. But I'm sure the one which we have heard a lot is something related to ABO or RH. Yes, you are correct. So the blood group which is most accepted worldwide is ABO type. Let's know that who discovered this ABO blood grouping. So it was Carl Landsteiner who discovered ABO blood grouping. So ABO system was introduced by this fellow Carl Landsteiner. He was an Austrian physician and he discovered ABO in the year 1900. And he won Nobel Prize for this in 1930. Let's understand how did Carl Landsteiner got to know about this ABO system. So let's go ahead. What did Carl Landsteiner noticed when he was working with these WBC? He noticed that on the RBC there are various antigens like antigen A or antigen B. So I'll be representing antigen A with a triangle and I'll be representing antigen B with a green color box. So on the RBC surface, if I have antigen A, I'll call that as A group. If I've got antigen B, I'll be calling it as B group. If I see both, I would say A, B. So that's about the Carl Landsteiner discovered on the RBC. But correspondingly, Carl Landsteiner noticed that in the serum, notice in the serum, what is serum? Serum is plasma minus clotting factor. So if मैं plasma में से clotting factors निकाल देता हूं, तो मुझे दिखाई देगा plasma, serum. So what did he observe in the plasma or serum? We say that he discovered corresponding antibody. Corresponding मतलब अगर RBC पे antigen A है, तो एंटीजन ए के लिए एंटीबॉडी ए नहीं होगा. What it will have? A corresponding antibody. Let's understand that. So he noticed that there are two corresponding antibodies. Like we got anti-A and we got anti-B antibodies. So where are these anti-A and anti-B antibodies found? They are found in the plasma. So for example, for the blood group with B will have which antibody in his serum or plasma? Anti-A. Whereas with the blood group A individual, what antibody he would have in his plasma or serum? Antibody B. So this is what he noticed. Now, based on presence or absence of these AB antigens, Carl Landsteiner could find out the following blood groups. Which four types of blood groups? Blood group A, blood group B, blood group AB and blood group O. So let me now conclude his learning by putting down a table for you. So here's the table. So on RBC, what do we find? We find antigens. And antigens are in the form of what? A and B. What do we find in the serum or plasma? In serum or plasma, we find corresponding antibodies. So let me put down four different types of blood group for you. So which four different blood groups are there? Blood group A, blood group B, blood group AB, and blood group O. Now, blood group A is determined by the presence of, yes, you guessed it correct, by the presence of antigen on the RBC surface. And as I told you, this would have a corresponding antibody. So, which antibody will a person with blood group A will have? He'll have an antibody B. Similarly, how do I identify a person with blood group B? So when I look at his RBC, I see that on his RBC, which antigen is present? It's antigen B and a corresponding antibody A. Now let's see what do we have next. It says AB. AB means on his RBC, both the antigens are present. Antigen A as well as antigen B, as you can see in the image. But what about the antibody? It will have no antibody corresponding for these because RBC bear both the antigens. So it will have no antibodies here. Is that clear? Now we come to blood group O. Blood group O means no antigens over the RBC surface with respect to blood group. So with respect to blood group, O blood group people on their RBC shows no antigen, means no A, no B antigen presented. So thus on RBCs of people with blood group O have no antigens on it. As they don't have antigens, in their serum or plasma we can have corresponding both the antibodies A and B. So this was about how did different types of blood group came into picture and what give rise to different types of blood group like A, B, AB and O. So I hope we answered our query that how did different types of blood group came into picture. So I'm sure you enjoyed this video. Please do subscribe, share with your friends ahead, and I'll be seeing you soon in the next video. Thank you all. Take care.